Good morning, good afternoon, everyone here in the room and elsewhere. You know, after that movie, it does remind you how much we're challenged to understand what life is really about, right? But we know we have to keep fighting, and so we're going to talk today about fighting because we're not giving up, and there's so much more to do. So I'll talk today about what I've been doing to try to change the future, and particularly in things we can do to try to make it easier to get these more advanced therapies that are coming forward into the clinic and also to approval. We have an opportunity now we haven't had in the decades where we have technology can get us there. And now we need to make sure our regulators understand what we're expecting of them and what it takes to get these drugs available and now being used in kids. And I hope we'll have it for others too. So the thing I've been talking about probably for 30 years since my first attempt to do development, which was, it turned out a drug we did get approved called Ultrazyme for MPS I. I learned very quickly the system can work and then sometimes can completely not work. And I wrote about that whole story, but because of that experience, I realized there's something fundamentally wrong with how things work. And the truth is, the drug development system isn't really designed to support development of rare diseases. There's approaches and standards and expectations that don't fit the kind of difficult, challenging disease we work on. And yet now we have these new technology that can actually open the door to treating things that we didn't have an opportunity to treat before. And so there's an opportunity really to get somewhere. But the paradigm, how it's done, the way the FDA thinks about it and other regulatory authorities, the sort of mindless attention to rule and pattern rather than looking at science, is that a fundamental problem. And when you have more difficult diseases like GM1 or other neurological lysosomal storage disorders, the paradigm doesn't work very well at all. And yet the precision medicine's here now, but we need those precision ways to measure disease and ways to determine, have I treated the disease, rather than doing what's traditionally done and expected for large market diseases, which are reversible and can be treated any time. When people tell me, how do I know when we'll fix the system? Well, I know when the system is fixed if families don't have to keep trying to develop their own treatment for the kid. As long as all these families are talking to me about trying to develop their own treatment, then you know something's wrong. That we have a scientific solution, yet families are having to raise the money and all the expertise that's out there not being applied, right? That's how you know the system isn't working. And that's how I'll know when it's fixed, because then that will stop and people will can be a parent and not have to learn how to be a scientist and drug developer too. So the Orphan Drug Act constantly cited and lauded 40 years ago. They had an idea and it has helped. It has helped, but the truth is that whatever that success is, it's just not enough. And it was designed at a time in the 80s. And the real point of the Orphan Drug Act was to take like a drug that had been developed, but let's say discontinued, and had to do with a particular drug at that time that was the focus of Abby Meyer. And that drug basically got abandoned or orphaned by its company. And so they needed a way to how to get that drug back on the market when there's no patents and there's no way to make it work financially. And that's the Orphan Drug Act was built around that. It wasn't really built around how to develop complex gene therapies for ultra rare diseases, right? That's not what it was built for. Now it did help. Look how many more things have happened. But I'll tell you one of the things you should notice like in the first 15 years, there really aren't very many treatments at all. We have thousands of untreated disease. We got a handful of actual approvals. Those are the red ones. The blue ones are designations. Those are all theories. The red ones are actually drugs. And then around the mid-2000s, it started picking up. And what actually was happening, to be clear and honest to you, is that Genzyme had a first treatment for a lysosomal called Gaucher. It was called Ceridase or Cerezyme. And they were charging a lot of money, and people were very angry at them. But they were able to earn enough money to help fund and do a lot of work. And once that happened, it actually opened the door to thinking maybe an ultra rare disease can be invested in. It's really important to understand, because if you have to spend $100 million, $200 million, who's going to give it to you? Who's going to give it to you? So in order to make it work, the fact that the prices were higher actually enabled this to happen. So I'm being real honest, that's what was started happening. And while people are upset about prices, I would also say to you that they are actually what helped this start, that we actually got going, because ultimately the money has to work. And you might ask where the money is coming from, but I want to be clear, it's not like Elon Musk or something, billionaires. The money actually, the majority of the money invested in biotech is from 401ks, 403bs. It's from retirement money, retirement plans. If you look at my company, I work at Ultragenyx. The top investors are all manage retirement money. So it's all of your money and all your friends' money that's in the bank. And so people are trying to invest your money for your retirement. That's who it is. That's where the money is. So we have to make the money work. And that's one thing I learned coming from being a professor and figuring out, how do I get there? The money's got to work. It's going to cost this much. We have to figure out how to get it done. How do we lower the cost? We're going to work that. But the truth is, Orphan Drug Act didn't solve the drug development problems. It solved certain aspects of things. That means there's a lot that's not done. And I think we know a lot more now than we did in the early 80s on what you need and how to do it. And that's what I'm going to talk about today. What we know right now is there's a lot of ultra-rare treatments. In fact, the last 3 or 4 or 5 years, there were a surge in even GM1 programs. There was a lot of excitement, a lot of optimism. Sanfilippo, some other very difficult diseases suddenly had a whole bunch of programs. But then the clinical development part got to be very difficult. The demands on them made it very hard, so it was near impossible. And gene therapies are very exciting, but they're also incredibly expensive to develop, very expensive to develop. And what's happened in the last while with some of the difficulties in the clinical development and the challenges from a regulatory standpoint, accepting easier pathways, we've had a lot of programs shut down. And we actually went and talked to the FDA about this very issue, that their behavior and their expectations and how they're managing things is hurting the investment and will result in a lot of possible treatments getting lost. And that's a tragedy. So the Orphan Drug Act itself is dated, not enough support. Now, one of the things people say about the regulation system, Orphan Drug Act is one piece, but people tell me, well, the FDA is flexible. The problem with flexibility is it's unpredictable. When are you going to be flexible? If you're only occasionally flexible and uncertain when you're flexible, or you're flexible in things that don't matter and not flexible on the things that absolutely are critical, it's not enough to create a consistent pattern of people investing in developing treatments and getting them approved. And so that way, you just hard to know what's going to happen. And particularly right now, I would say that's one of the challenges. People don't know if they're going to be flexible on some endpoint or trial design, or they're not. And if you're in the middle of it, what's happened, people have discovered what they're expecting of us is going to take so long and so much money that we have to cancel. And that's a tragedy. Now, the other thing that's not accommodated for very well at all is the fact that the disease is very complicated. All right. Factor VIII has a lot of treatments, hemophilia A. Well, it's because it's in your blood, it's a factor, you bleed. It's very simple, right? It's for one compartment disease. It doesn't affect everything. As soon as you get into diseases that affect multiple organ systems, multiple body parts, chronic, complicated, the system is not designed for that. It's designed for diseases that have like one symptom and you measure them. When you have multiple symptoms and it's complex, that's tricky. And even more complex when you're talking about brain disease is the fact it can be irreversible or variably irreversible, as well as variably manifested. So not every kid that looks the same is actually the same inside. There's different stages inside their head of what's going on. And that irreversibility creates a lot of challenge in trying to do clinical endpoints, particularly in diseases where by the time you know what they have, it may be too late to actually reverse the course. Some diseases can be reversed, some partially reversed, some not at all. So the system doesn't work for that, because how do you do a clinical trial with a clinical endpoint when you have to treat a patient before he has clinical symptoms? What are you measuring? How do you measure? And if you can't make things better, only stabilize, then how do you know that they're better without waiting a very long time? So the variability and this irreversible and the long time scales make the system's not designed for that. And finally, the other way then of solving the problem would be what's called accelerated approval. That's where you measure a biomarker, like the substrate that builds up in GM1, measuring that and saying how much of that's there. The FDA looks at those as a compromise. They look at it as not really the disease, but I'm here to tell you that's wrong. The storage material, the stuff that builds up, that is the disease. What happens to you in cognitive function and walking or other features, those are outcomes. That's what happens to you because of the disease. The disease is the substrate building up. It's a biochemical disease. The outcomes what happen to you later. I guarantee you, if we focus on treating the underlying disease, the cause, we will have a better answer to what's going on than trying to use a clinical outcome, which is very noisy and very downstream from what's going on. And this is a fundamental problem with the change in the definition of disease to not be the clinical outcomes, but to be the underlying cause. If we can do that, what we're talking about accelerated approval is measuring the underlying cause, not the outcomes of disease. And this change in mindset is a way to change and move us forward. And I'll put this to you that the best example of this is HIV, human immunodeficiency virus, HIV. That disease causes, let's say, problems with immune function, infections, hospitalizations, death. But that process can take 7, 8, 9, 10 years to get there. But the disease is the virus. The outcomes are immunodeficiency. So if you demanded clinical outcomes to do a trial, it would take you multiple years and very large trials to measure loss of immune function, because it takes so many years for that to happen. The truth is, the reason HIV is in a better place today is because accelerated approval said, we're going to let you use immune function in the early measure, or ultimately how much virus is in your blood. That's going to be the measure. But how much virus in your blood, that is the disease. Do you have virus in your body? And when they developed drugs that way, within a 16-year period, they had 29 drugs developed, 4 combinations, and highly active retroviral therapy, because each drug could be added and a second drug added, and very quickly, within a trial of only 8 to 10 weeks long, figure out if you're better. The cycle time was very quick. We changed the whole story. And I would guarantee you, if the FDA had prevented accelerated approval from coming forward, because they were resistant, they thought it was a mistake, they thought it was going to ruin drug development. If they had prevented it, we would not have had this solution. The truth is, measuring the underlying disease is the correct way to develop the drug. A drug that actually treats the cause and not just manages symptoms. But the current system focuses on managing symptoms and downstream clinical outcomes, not treating the underlying cause. I make this point to you to also begin the process of changing the mindset of what disease is. Disease is the underlying cause of disease, and the rest of it are outcomes. If we do that, we can change the way we think about all of it. Now, in GM1, there have been, I'm sure for Christina, big surge in excitement, a lot of programs coming in, then things got canceled. There's still possibilities there, and we're happy that Passage is still working it. I think there's great hope for the gene therapy in GM1. I think it's working. I think that there's great hope, but there is challenges. How do you get to the end? How do you get to an approval? If it takes forever in doing clinical endpoints, I guarantee it will be very, very hard. This is one where biomarkers need to be accepted. Otherwise, we'll end up not getting to where we could with the opportunity we have in front of us. Another example I often bring up, because I worked in the MPS disorders, or mucopolysaccharidoses. These are variable lysosomal storage disorders as well, but if you look at the approvals we've had in this disease, because there have been 5 enzyme approvals, they all treat the body part, not the CNS part. And CNS-directed treatments, it's been a lot harder. For MPS 1, there's been at least a dozen companies working on improvements. The first treatment was approved in 2003. So 20 years have passed, not a single follow-up drug, not a single improvement, and none affect the brain. It's not an accident. The original approval, we were forced to use clinical endpoints instead of biomarker, which made it very difficult to develop the improvement. And without a biomarker for the CNS, it's extremely hard to get enough patients and to do a trial. And so we've had some treatments, I think, that were improvements not get approved. Now there's some other things coming, But if you look at not only MPS 1, but MPS 3, which is called Sanfilippo, there's a lot of complex, irreversible disease. The timing and treatment is narrow. The time courses, they seem fast for a family, but from a drug development standpoint, taking 2 or 3 years is incredibly long to try to look at and run a trial. And the question is, does it make sense to have a control group that's going to lose its opportunity to ever be treated? Do we have to sacrifice people's children in order to figure out how to science? Are we not good enough scientists to figure out an answer without having to sacrifice lives? I think these are fundamental questions. So traditional trials, I think, are very hard to do, and we have to get better at using the biomarkers, understanding what they mean, how they measure underlying disease, and figure out a way forward. And similarly, in the MPS disorder, Sanfilippo had a period where there were no treatments, and then there was, look at all these companies, a whole bunch of programs. And now you see all the ones that are red, those are all stopped, and others at risk. So this great surge of activity is now turned around because the biomarkers are not getting accepted and things are challenging. And at Ultragenyx, there was a program from Abiona for MPS 3A, and it was working, but they couldn't afford to finish. They couldn't afford it because FDA was asking them to make every child go till 5 years old. They have to be 5 years old, but you have to treat before age 2, right? So at least 3 or 4 years of follow-up in order to figure out whether they were being treated or not. And they just couldn't afford to do it. They dropped it and they just called me up and said, look, we'll give it to you if you'll take it. Someone's got to save this. And we opted to save it, even though we already were at our limit, what we could do. We are working on it, but part of our goal now is to change the system and to use that program to help change the system about how we approach biomarkers. So we know proving efficacy in ultra-rare disease can be difficult. Clinical endpoints, sure, they're better if you can do them, but a lot of the time you can't. Powering randomized trials is extremely hard to get enough patients to deal with a neurological symptom. Most neurology drugs have several hundred people in them, right, that treat seizures or treat other types. You're never going to conduct a trial with that many people, so you can't power it and you can't deal with the high variability that exists. So that's very hard. Natural history can work, but the problem with natural history is slow, that the FDA hasn't really accepted that control very often. The only time it has is when the natural history is very fast, like in Pompe disease or in late infantile Batten's disease, where the decline goes essentially from normal to near death in 2 years, right? When it's that fast, then they will have accepted the control group. So the idea of using natural history has been challenging. It should be acceptable, but the number of rules and debate that goes on in dealing with the agency, they're so afraid. And I'm thinking, why are you so afraid of some imperfection in the natural history data? You should be more afraid of letting a treatment that could work not get applied and to stop the development. That should be a bigger issue, but they're more afraid of accidentally letting something imperfect. And I think that's a mistake because they're not lining up to do what we want them to do. It becomes actually, you would think it's easier for gene therapy or single shot therapy, but it's actually worse because even though the therapies look expensive, the amount of money spent on them is so much higher. And therefore the truth is that actually becomes much harder to be able to make the financial part work. And so with gene therapy, it's even more imperative that we have more efficient ways of doing the drug development. And we just need to be able to qualify biomarkers, which means proving the FDA or regulators that what this thing we're measuring is a reasonable way to assess the disease. So we know the science, though, is really strong. And just like in GM1, you have a block. We know what the enzyme is. We know what the enzyme does. We know what builds up. There's just no mystery. It's all known. It's very distinct, very clear. It's not like biomarker for a common disease that you randomly pick a blood test. This stuff is the material building up. We know exactly what it is. Not surprisingly, if you look back in time, in the 90s, the FDA had approved a number of rare disease drugs using biomarkers. At that time, they didn't demand a lot of stuff. They were just going off the science. That's when I first went to them. They said, look, scientifically, it made sense. They let us move ahead. And then someone got the big idea and said, no, we need to have very high standards for this. And they changed. Why, I don't know, but because if you look at the drugs, including about 30, they all work. They all work. And the biomarkers have been used repeatedly, correctly, like ammonia for urea cycle defects. But I'm now dealing with another urea cycle gene therapy, and FDA says, I'm not sure I believe ammonia is a primary endpoint. So you have 8 or 10 drugs approved with it. It clearly is the cause of disease. Now, suddenly, we have to retest the theory. That is a mistake, but that's what's going on. The truth is these biomarkers that measure underlying disease are very accurate. And I will guarantee if you want to try to improve, they're the way you're going to be able to make an improvement. If the ammonia control is pretty good, then making it better, that'll be a better drug. By improving with these biomarkers, we're actually open the door to rapid evolution and improvement in our treatments. That's how we get to the end. We got to change the system, just like it was for HIV or other, like Hep C. Now, there's other things going on politically that are creating a problem. The recent accelerated approval of an Alzheimer's drug and the whole political debate, how that went down, brought out people like Public Citizen, who, I don't know why they think of them public citizen. It's really a particular political group that thinks they represent everyone. And you have all these people attacking accelerated approval as being, a false approval or not really good drugs. And all this debate is just basically wasting time and obstructing. And it has nothing to do with the ultra rare diseases, right? Recently, there's been bigger demand for, I think, unethical randomized trials. In a Sanfilippo program, they're asked for the control group could have at least 15, have up to 15 to 20% brain damage as acceptable, and then they could come off placebo. What the FDA didn't really indicate, I'm telling you today, that in order for the trial to work, the control group has to have the brain damage. That's how they're going to separate from the treated group. You can't see a treatment to show the drug works unless the control group has declined. If they don't decline, so it's not a maybe if they decline, if they have to decline. So you have to go long enough in order for the control group. But what's dumber about that, completely idiotic, is the truth is with Sanfilippo that the time it takes to have that happen, you've lost your window to be treated. And you will not recover because you now lost the window, you're past the 2-year point. And at that point, you have no chance of reversal. You're going to die. Is that the trial design we all asked FDA to do? Really? That is just a travesty. And I had someone from them see, it's not too bad, it's just a little bit of brain damage. And I'm thinking, I imagine if you were a parent with some of these, you would be grabbing that person by the throat to have you tell you that's okay. It's okay for someone else's kid, apparently. How do we tolerate that? How do we ever let them say something so egregious? We can do better than that. And there are ways to prove drugs with solid science. They don't have to do that kind of a ridiculous thing. The other problem that's happening at FDA is there's a lot of turnover. I'm on meetings with FDA now where the reviewer has like 1 year experience, and the most senior person in the room is 4 or 5 years. And they don't train them because they don't know a single drug that's been approved before, even in the disease area that you're talking to them. They don't even know. You're talking about MPS disease. They don't know about the other MPS approvals. They don't know anything about them. And what happens when you don't know much? You get rigid, right? You go by your training. You're not able to think because you're afraid. And so you have these people that are afraid, that stick to rules and are rigid. And the only way we survive is we try to reach someone more senior to FDA to intervene and use some rational good sense who actually knows something. But how often can you keep doing that? It is hard. So I'll tell you, there is a challenge. Now there's also another problem with threats against reimbursed drugs, that once it gets approved, that reimbursement is saying, hey, that's not really a real approval. It's like, well, one thing after another. While this seems very dire, and I'm being a little bit strong here, it's not. Technology's there. We got options. We just have to do something. I'm currently working on a campaign to raise awareness about the need for biomarker-based approvals, and we're going to go hardcore with communications, lobbying, and on the Hill. And we're also going to engage FDA directly in workshops on the science side. But the long haul, I think we need to change. And I think the Orphan Drug Act has been good. It sort of accidentally worked out. But the Ultra-Orphan Drug Act, I think, is something we're going to need. The idea here is pretty simple. Orphan Drug Act has for 200,000 patients in the United States. Well, 200,000 is like some gigantic number, right? Compared to all the ones we're thinking about. I think if we talk about 2,000 identified patients in the US, there's a whole lot of diseases in there where there's good science that are totally, it's a very small population. It's 1% of the Orphan Drug Act. And the idea here is politically to talk about this is the 1%. The 1% where we have the science to fix them, and we need to make the rules special for that 1%. We need to change how the efficacy standard, instead of getting substantial efficacy, which FDA's mind is 2 randomized trials and all this stuff. We need to put what I call the sufficiency efficacy standard, similar to what's used for the emergency use authorization for a drug, like COVID vaccine. The emergency authorization said it has to be sufficiently efficacious. If we set that standard, it changes the rules of how they behave and how they would interpret. And the FDA does respond to the rules and the law. We need to improve the ability to qualify biomarkers, primary disease biomarkers of a certain type, and I think that needs to be codified so that they cannot require long-term clinical outcome data to prove the biomarker when such data is always impossible or impractical to collect. I also think for these ultra rares, we need the 50% tax credit for orphan drug to come back, but we cap it at 100 million, which means for the really small ones, there's a better incentive. If you're investing that much money more, then it's clearly not that small. You have a potential return. But if you had a better tax credit, it might help the financial story that would help some of these things get done on the really little ones. It would help the little companies that need a break to make this all work. And finally, I think the approvals shouldn't be called accelerated approval. They should just be called approvals. That doesn't mean there shouldn't be a post-marketing commitment. They should be. They should be allowed to have a post-marketing commitment where you verify in the long run your drug's doing what you think. That every company should be held to the standard of assuring they've done what they can do and figure out what's not right to set the stage for improvements. And everything like this will take step-by-step improvements will come. So how rare? I've set 2,000. You might, for GM1, I think you're in good shape. I guarantee you there's some rare disease people are going to say, well, our disease is 5,000. What are you doing? But the truth is, as you get bigger, it's a better financial situation. If you're bigger, there's a potential that makes it work better. And my view is that the less than 2,000 one, they need the help, and politically better to keep it tight. That's why we focus there. We think that's the greatest help. My hope is whatever we do for the smallest group will change how the FDA behaves for others too, right? Think of it as opening the door to change, and now a pattern gets set, and now that will alter, I think, the way others might get treated. So my sense is keep it tight for the political reasons and the ones where we really need the most help. So time for change, enact an Ultra-Orphan Drug Act. Now, this is something that takes a couple years. Our campaign to get biomarkers qualified is going on right now, and we might need your help at some point, and I'll reach out to Christine when it happens. She was with me when we went to FDA. But the longer haul, 2 or 3 year plan would be to get an Ultra-Orphan Drug Act to codify the changes just as I would describe today. And we think that a new Ultra-Orphan Drug Act with these changes would re-energize investment in some of the smallest companies out there, which would allow them to actually get to the next level. And it would really focus the incentives now on the cases where the disease are really small and they really need the help, and where our science is actually ready to do great things. This is a place where I think we can do great good, and our hope by doing this is we'd fix the system and change how it's done for ultra-orphan rare diseases. I did include some references for Christine in the PowerPoint deck if she wants to distribute them to talk about some of these topics. But I do believe there's a lot to do. I believe that GM1 is at a place where it can get treated, and I think it can. But you have to be vigilant. You'll have to fight and support politically with FDA to make sure the right things get done and these treatments get to the end, become available. Thank you.